Assalamualaikum warahmatullahi wabarakatuh. Thank you, Prof. Ravi, for the kind introduction. So, when we talk about genetic medicine, we have to admit that most of the applications of genetic medicine nowadays really centers around penyakit yang jarang dijumpai, rare diseases, or another term for that is orphan diseases. Why orphan? Because everyone neglects that. So everyone, from the perhaps from the patients to the researchers who are working on that field, were practically perhaps neglected. So what I would like to make a case, argue in this lecture, is that what we have perceived as rare is actually not rare. These are the definitions of rare worldwide. So every country have their own definition of rare. When the disease hits below this frequency, this prevalence in the population, then it is regarded as rare. This is the individual cases. Like we can see, for example, disease like spinal muscular atrophy, which happens only in 1 in 10,000 or 1 in 15,000, and then the Duchenne muscular dystrophy, which happens only 1 in 3,500 males, things like that. Then Australia perhaps strikingly higher, put up the higher numbers because of the low populations itself, perhaps. But then, when we see actually how many people around the world have the rare disorders itself, it's not rare. It's not. This is not the number that we can really put aside, right? 350 million people worldwide have the disease, have rare disorders of any sort. We have thousands of rare disorders, and if we combine together patients who suffer from all the so-called rare disorders, with the prevalence of 1 in 10,000 or 1 in 6,000 or something like that, so we come up with this number, 350 million. This is the whole Indonesian population. This is the 14 times of Malaysian populations. You see? So it's actually, the number is quite exaggerating. And then how many children were born every year with the disorders? It's also so many, 7 million. Right? And sadly, then 90% of them are born in developing countries. And then when we compare that with the so-called common disorders, this is what the figure from Australia, which turned out not to be strikingly different. See, 1.2 million people in Australia suffer from rare diseases, which is the same number of Australians with diabetes. Malaysian figures I have in later slides. You will be also strikingly surprised when you see the numbers. And this is the sad thing, is that 95% of rare diseases doesn't have any FDA. FDA is a Food and Drug Administration. It's a drug authority in the United States that regulates drugs and release license for drugs. 95% of those kind of diseases doesn't have any FDA-approved drugs, FDA-approved curative treatment. Most of them have treatments of some sorts, but almost none of them have curative treatment yang boleh menyembuhkan, treatment that can cure. Now, what do we have in Malaysia? 56 out of 1,000 babies in Malaysia were born with birth defects. So it's not trivial number, of course. And then each year there are about 20,000 Malaysian babies born with birth defects. And then based on extrapolated data, so this one is obtained from the assumption that 10% of populations in any country, they will suffer from any kind of birth defects or any kind of rare disorders. Then we have 2.1 million Malaysians live with rare diseases. And then how many people in Malaysia live with diabetes? With the prevalence in 2007, we have only 1.9 million people live with diabetes. So this is not rare. This is clearly not rare. This is clearly a burden. This is clearly a problem to a nation. Right? What's the problem then with why do we look at why do we perceive that as rare? This is because of first of all, there is a false perception that the disease may not be important enough. I had experience of applying grants, of course, for one type of rare disorders, finding cures, finding treatments. The first questions they give to me is that how many patients do you have with that disorders? I come up with 30. Okay, all right. So you are applying this million of ringgit Malaysia for 30 patients. So what can I say, right? But I would argue, of course, that the treatment that we can get from this study may be applicable to other diseases. And then also there is what are other problems with rare diseases is the way people approach it because of the absence of cure and treatment. So most treatment will be supportive and not curative. And then low knowledge among I have to admit, among health professionals with regards to this kind of diseases, and then also the public. And then this one, the fourth point I would like to stress further in the next slides, that we have kind of fragmented perspectives. So we don't see rare diseases as one entity, but rather we see rare diseases in each of them. How many DMD patients do you have? How many SMA patients do you have? How many Down syndrome patients do you have? How many Tay-Sachs patients do you have? Neurofibromatosis and all. So fragmented. Then people have reasons why they see it in fragmented views. That is because perhaps the root diseases are also different, and the way treatment is done also may be different. The problems that they have may be also different. But what I'd like to emphasize in this today's lectures perhaps is we can see them as one entity, especially when we study it, especially when we propose a certain treatment modulation study. This is why we can see it as one entity. So, in fact, actually 80% of thousands of rare diseases, they have genetic origin. They're rooted in genetics. Some of them are congenital abnormalities, which are caused by infections during pregnancy, for example. But it doesn't make up most of the types of rare diseases. In fact, 80% of rare diseases have genetic in origin. So, with this information, then actually we have common views on how we see the disease with regards to understanding the principles in the pathophysiology. When we study the disease, maybe because when we study genetics, when we study molecular biology, whoever, from students to professors, I think later in the lectures perhaps we can see it. We will only work with one dogma, central dogma of molecular biology, from DNA, and then DNA transcribed into RNA, RNA then translated into proteins. That's it. It's not more than that. Right? And then with 80% rare diseases having genetic in origin, then we should have common perceptions. We should have common principles in understanding most the mechanism, the pathophysiology of most of the diseases. And then because 80% of rare diseases have genetics in origin, then in terms of markers for diagnostics and for prognostics, we also have something in common, which are also genetics. 80% of them have problems in the sequence of the DNA, have error in the sequence of the DNA, deletion of some segment of DNA, changes in the DNA sequence, and so on. And finally, when we propose a therapy, we will also talk about the same components, genetic components. When we target genetic components, then we talk about 80% of the rare diseases. Right? So now I'll illustrate one of the example of how just small fraction of study in genetics, they can address a quite significant portion of disorders in genetic. Splicing is probably splicing is one step in how you translate or transcribe from DNA into protein. We have DNA in our nucleus, in our core of the cell. So that DNA is actually a code which is beneficial for the work of that particular cell, right? And then in order for that DNA to act as a code, there are some processes which is called splicing. Not all part of the DNA will be used to translate protein. No. Some part of the DNA will be used and some other part will be removed. So the process of assembling the part that will be used as protein to translate for protein, and the removal of the other parts that will not be used, that process is called splicing. So when there is an error, which part will be used and which part will not be used, that error can cause disease. Right? So then when we address that process, actually there are quite a significant number of patients with certain diseases have these problems. 50% of patients with neurofibromatosis have splicing problems. 50% of patients with mutated ATM have problems with splicing. And 96% of patients with SMA have problems in splicing. And we target that. If we target that process, the splicing process, we might be able to address a many number of patients and perhaps also many number of disorders. And then actually 60% of genes in chromosome 22, for example, are represented by 2 or more transcripts. What does it mean? 2 or more transcripts. We have a stretch of DNA, and the molecular machinery have the system on how to recognize which one to be assembled and which one to be removed. This can happen as an alternative. I mean by alternative is that one version can be that perhaps 200 code will be assembled together and made up into protein. Another version can be 250 codes assembled together and made up into protein. And both of them can be an alternative version, and both of them can be normals. So this mechanism underlies what so-called splicing alternative. Alternative in assembling which one to be put up into protein, which one to be removed. So actually, quite a significant number of codes in our cells are worked up in that way, in the splicing alternative. And then, this is another effort of generalizing this impact of splicing in the human diseases. 15% of point mutations that result in human genetic disease cause RNA splicing defects. So RNA splicing defects, splicing defects in picking up which part to be assembled together and which part to be removed. Error in that process cause 15% of made up of 15% of mutations. And this is only splicing, and we have many number of processes in molecular genetics. We have translation process, we have transcription process, we have epigenetics process, many things, and each of them, they have their diseases. Many different diseases, they may have they may grouped into disorders of translation, disorders of epigenetics, disorders of transcription, like that. Then I'll come up into just two examples of rare diseases. First will be Duchenne, the so-called Duchenne muscular dystrophy. This is an inherited muscular dystrophy. Muscular dystrophy means broken down of the breaking down of muscles, because of the inability of muscle cells to retain its integrity. Then over the years, throughout the life, then there are many numbers of cells broken down. When there are so many numbers of cells broken down, then numbers of cells building up, then we have we can see disease manifest. There are many types of muscular dystrophy. One of the type is the so-called Duchenne muscular dystrophy. This is progressive muscle weakness. We can see from the video that I'm going to display in a moment how progressive it is throughout the childhood life of humans. X-linked, we have seen Dr. Lind making case about X-linked recessive disorders, which means that the disease, the disorder will be in the X, and then because females have 2X, most females will not be affected, and most patients who are affected will be males. Happens in not so rare actually, 1 in 3,500 males. This will lead to reduced life expectancy and quality of life. We'll see in a moment how the disorder progress in the life throughout lifetime. There's no sounds, because we can read from this one. So from 8 months, we may not see any differences from other babies. Then, by 3 years of age, we started seeing some problems. So this is a like early sign of the so-called Jawor's sign, where it takes effort for the baby to get up from sitting position. We see they're having problem even to step up to the stairs. By 6 year, you see like a very characteristic gait, the way the patient walk, it's very characteristic. So it denotes difficulty in walking. Then of course with that gait, with the difficulty in walking, then it's difficult for them to keep up with their normal friends. So can still walk, can even still run, but of course cannot keep up with the other peers. See, by 8 years, then almost cannot stand up. It's very much effort to stand up. This is a very characteristic of the so-called Jower syndrome. This is how the doctors usually recognize the pattern, the clinical symptoms of this kind of patients. This is describing the efforts to get up from the sitting position to really standing up. Right. Then by 10, it's no longer possible to walk. All right. So this is soon after that. Then most of the muscles, including what was affected first, will be the limb, the limbs, and then the arms will be later affected. Most of the time, that's that will be the problems. All right. Then what caused this disorder is the mutations that happens along a gene called dystrophin. Dystrophin is the largest gene in human, the biggest one. It's located in X chromosome. 3 million codes of DNA, but it is translated into a very tiny 14 kilobases of RNA. So the mutations that usually occur is the deletion, the removal of a certain portion of coding DNA along the gene. And what dystrophin do is this is dystrophin. Dystrophin is like a rod that acts like an anchor that assemble together parts of the intracellular muscles with the laminin, with the cytoskeleton. So if the anchor is broken down, then you will have the muscles disintegrated. That's very logic. And there are also variants of the disease where dystrophin get mutation, but it's not losing a total function of it. The dystrophin, the rod one, the anchor, is just getting shorter. That will give disease manifestation, but much less than that. And in fact, this is how currently therapeutic study was targeting at. Okay, we don't have the whole dystrophin. In DMD, we have the dystrophin, all of the dystrophin, we get it disrupted. But one of the current therapeutic study is that, all right, we cannot restore the whole dystrophin, but we can perhaps make a shorter version of dystrophin. So it's not totally curing it, but perhaps very much reducing the severity of the disorder. Another one is I would like to touch on spinal muscular atrophy. This is the study that I study from. I start my introduction with molecular biology studying this gene. It was 2003 back there in Japan. Different from muscular dystrophy. In the muscular dystrophy, the problems lies in the muscle itself, the breaking down of muscle cells. But in spinal muscular atrophy, as the name denotes, spinal and muscular, the underlying pathogenesis happens in the spine, along the spine. The loss of motor neuron, the loss of neurons. Neurons is cell sharaf, cells, neural cells, right? In the spine, because there's innervation, because of the loss of motor neuron in the spine is lost, then the ability to control the limbs also lost. Then you won't be able to walk, and also you won't be able to function the muscles that rooted from the spines. Right? In a very severe form, the severity is variable from type 1 to type 4. Type 1 is extremely severe. You were born and then you got the disorder. The first characteristic was the inability to properly breathe, to sufficiently breathe. So most of the type 1 patients died within 2 years of age because of the inability to breathe. And then we got more and more, less and less severe as the type goes up. This is autosomal recessive, as Dr. Lin has mentioned, autosomal recessive, and occurs quite rarely, in 1 in 10,000, 1 in 15,000, and also lead to reduced life expectancy and quality of life of the patients. So I think I'm not going to put this one, just a little bit of this. This is a video of no. Okay, it doesn't work. All right. Okay, it was just actually a video of somebody named Mikita from United States. He is assistant to the Attorney General in Utah, suffering from SMA. So we can see that people with SMA, they can excel. Their cognitive function is intact, and then of course then they can go up to university with this is how the disease occurs in terms of pathophysiology. In chromosome 5, we know there is a locus called SMA locus. This is the so-called SMA locus, the locus of spinal muscular atrophy that exists in this one part of chromosome 5. Right? 95 to 96% of patients, they have deletion in the gene so-called SMN1. But within that locus, we also have other genes. The most important of these three, except SMN1 because it's already been deleted, is SMN2. Why? Because SMN2 is actually the twin gene of SMN1. But because of some difference, because of very little differences in SMN2, you see, the logic is that if we have twin gene, this one is the same with SMN2. the logic is that if we have this one deleted, if we have the twin, then we should be this one should be able to compensate. But because of the little difference in here, the problem happens in how the cell can use the SMN2. So the cell cannot use properly SMN2 because of little change, little difference between SMN1 and SMN2. So actually we have a twin gene. So this is right. This is the SMN2 gene is a twin gene. Although normally without any intervention, SMN2 gene cannot really function, but with some interventions, which is now how most therapeutic study is targeted, is to enhance the function of SMN2, so that SMN2 can, to a significant extent, replace the SMN1, then compensate for the loss of SMN1. Right. So I guess this will be the last part of my talk today. Then with this group of disorders, we should see impartially when we have a scientific look. So we study transcription, for example, or we study splicing, for example. That will be not only for a certain disease, but also for a many group of diseases, which can target many patients. And then, yes, therapeutic studies can generate a lot of money in the results, but you won't go to therapeutic studies if you don't really understand the disorder. Most of the time, you have to understand the disorder, you identify the target of therapy, and then you start studying the therapy. And then, this is what I am getting more and more excited. Look at our own backyard. We have so many natural substances which grows in our own backyard, and many of them actually have effects towards molecular processes, towards modulating splicing, for example, towards modulating transcription, for example, which can lead to cures, which can lead to disease to improvement. And no doubt we have to join forces like what we do today, partnership between the academicians and then the NGOs and the public, and also patients gather together, gather the strength for the betterment of the patients. In the United States, there is a so-called Orphan Drug Act that give benefits, incentives to pharmaceutical companies that produce drug for rare diseases. And then the last, public awareness, of course, in this kind of disorders, philanthropic efforts, partnership between the government, NGOs, and the private sectors. With that, I guess I would end my talk today. Thank you very much, and then we can discuss more in the coming session. Thank you. Assalamualaikum warahmatullahi wabarakatuh.